I'm going to take this a little bit different to some of the other talks and try and go through quite slowly and explain some of the key concepts as we go through. As we know, we've heard a little bit already from some of the talks about antibodies, and vaccines are a second form of immunotherapy, which we heard about yesterday. But I'm going to talk a little bit about cell therapies, the concept being that we actually take a patient's cells or a donor's cells and inject them, manipulate them in a way to attack or recognize cancer, and then inject them back into that patient. Now, T cells are the cell I'm going to focus on, and these are the predominant type of cell within our bloodstream for recognizing something as foreign. So it could be an infection like a virus, or it could be something like cancer. And T cells have some potential advantages for these kind of therapies because they have a natural ability to migrate into tumors. If you look at a tumor, you can actually find large numbers of T cells infiltrated into it. Cancers are actually foreign to our immune system because they undergo so many changes, and there are various things on the surface of cancer cells which are naturally recognized as foreign. The immune system always makes an attempt to eliminate cancers, and actually the development of a cancer is a failure of that process of elimination, usually caused by the cancer evolving some mechanisms of hiding itself from the immune system. So actually the key is trying to understand those mechanisms and combat them. In other words, revert the balance in favor of the immune system and away from the cancer. T cells can kill cancers directly through various different mechanisms. And one of the most attractive aspects of it is just in the same way that once you have immunity against a virus, you often have lifelong immunity. If we can so manipulate T cells to recognize a cancer as foreign, then potential exists for lifelong immunity against that cancer. Of course, people have been saying this for 20, 30, 40 years. There's nothing new in this concept. But there is now emerging evidence that the technology has evolved to such an extent that this is becoming more of a reality. One of the other advantages of T cells is they actually recruit other cells to the side of the cancer. Now, this is a cartoon showing the concept of what we call adoptive immunotherapy. That means taking cells, manipulating them outside the body in some way, selecting for the green ones, which are the ones which are going to recognize the cancer as foreign, and then injecting them back into the bloodstream so that they will go to the tumor and effect a killing of the tumor. Now, the next concept I want to talk about is viruses, because we actually use viruses in this technology. Now, all a virus actually is a DNA molecule. It's a gene or a little collection of genes which are capable of making copies of themselves. And there's lots of different viruses which do this function in different ways. The principle is they have on their outsides these blue things, which are little proteins which are able to effect entry into a cell. And within them, they have the DNA, which encodes more proteins, which enable them to make more viruses. It's very, very complex, but that's the principle of how viruses work. And the reason I introduced the concept of viruses is because we can now engineer viruses so that they will have new genes in them which we want to put into a cell. So viruses are one of the main ways of genetic engineering or genetically modifying a cell to change its function into a function which we actually want it to have. So without going into the details of how you do it, we're putting in the red gene, you can see at the bottom, which is the gene we want to put in the cell. If we imagine down at bottom right, we're talking about a T cell, so the killer cell of the immune system. The virus can infect the cell, put a new gene into the T cell. That new gene gives the T cell the new function. As a result of that new function, in theory, it acquires the ability to recognize and kill cancer specifically. It's not a new concept. It's been around for many years. Now, one of the advantages of this is if the virus gets successfully into the cell and puts the new gene in, which is shown as the blue thing in the chromosome there, when that T cell divides and makes copies of itself, each new copy also has the new gene in. So if we just put a small number of T cells with specificity for the cancer in, and we can trigger them to divide and make copies of themselves, we can end up with a very large population of T cells within our body, all of which have the same specificity for cancer. So I think that's what we talk about, a memory response and creating lifelong memory. for the tumour. So now the concept of adoptive immunotherapy is this. We take blood from the patient, we extract the T cells in the lab, we add the virus to the T cells, so we then select for the T cells which have been engineered to now recognize the cancer as foreign, and those are the cells which are specifically injected back into the patient. And hopefully will migrate to the tumour with more activity than standard T cells will, and bring about the killing of the tumour. So we're now going to talk about CARs, going from one technology to another. The reason we're going to talk about CARs is because I've talked about the genetic manipulation of the cells. So what is the gene we have to put into the T cell in order to have this magical effect? And the answer is it's a CAR gene, so-called because it stands for chimeric antigen receptor. So nothing to do with these kind of CARs, but to do with the bottom right type of CARs. Now this is the most technical slide in my presentation, and I won't go through all the detail of it, but I will try and explain the concept using Daniel's pointer. If I can operate the technology, which, oh yes, I can. In fact, you can't see that, so I won't bother. So on the left is what we call an antibody. We've talked about antibodies already. And this, this part of it here is the bit which binds antigen, so the thing which is foreign. An antigen is the thing which is foreign. And antibodies float around in our bloodstream and specifically bind to things which are foreign. So GD2 is the antigen which many of us in this room know about. Now the thing I've drawn in the middle here is a very complicated molecule present on T cells, which is the way in which T cells recognize things as foreign. And the important part of it is the orange bit, which is the part of that molecule which tells the T cell to kill what it has recognized. So chimeric means you make a mixture of two different things. And a chimeric antigen receptor, or CAR, therefore is chimeric in that it's got the, at the bottom, top right, it's got the antigen binding part of an antibody, and at far right at the bottom, it's got the orange part, which is the bit in the T cell, which tells a T cell to kill what it has recognized. So that's the concept of how a CAR works. Now, we can therefore adapt this for neuroblastoma by getting our CARs to be CARs which are specific for the antigen in neuroblastoma. So it could be many different possible antigens, and an antigen for neuroblastoma is something which is present on all neuroblastoma cells on their cell surface, but is not present on many normal cells of the body, because we don't want to kill normal cells. So I've drawn the chemical structure, as you can see here, of GD2 underneath our neuroblastoma cell, which is a small molecule nearly always present on the surface. And in our CAR, we now have the antigen binding part of our antibody and our T cell signaling at the bottom. So the CAR is what we're going to use in our genetic engineering to make our T cells specific for neuroblastoma. So to reiterate, we take our blood, we engineer it with a virus. It now expresses the CAR on the cell surface, which has specificity for GD2, and this is what gets administered back to the patient. That's the concept, and it's not a particularly new concept. Now, as I say, adoptive immunotherapy as a principle has been around for at least 20 years. And some of the earliest successes which show that in principle this could work came in melanoma using different technologies. And in melanoma, it's possible to take T cells from a patient and inject them back in after growing them up in the lab. And it's known that you can get not just these kind of dramatic clinical responses, but these can be lifelong immunity induced. But those successes in melanoma have never been translated into other cancer types using this, what you might call old-fashioned technology, which is not to say this is not a great advance in melanoma. So, some spectacular results in recent years in leukemia. Can this be translated into neuroblastoma and solid tumours is one of the key areas of interest in the field. Now, the initial trials on GD2 CAR, therapy were held in the States in the 2000s and were published in 2006 and 2008 respectively. In 2006, it was a CAR in just a very small numbers of patients recognizing an antigen called L1-CAM. 2008, it was a CAR which recognized GD2. I'll show you briefly some of the results from these trials. So the initial trial in Baylor went on to recruit eventually a total of 19 patients, in whom there were 3 complete remissions, and just one of those is shown there. And there was a reasonable survival of the T cells. In the right-hand side, you can see one of the publications which has come out in the last 2 to 3 years, targeting leukemia, using a different type of CAR, in which the results have been more spectacular. So indeed, in the Children's Hospital Philadelphia trial, which was published this year, in their 30 patients with chemotherapy-resistant leukemia, they got 27 complete responses out of 30, which is really an unprecedented success in any childhood cancer for children who have now become resistant to conventional chemotherapy. So as a proof of principle, it shows it's potentially exciting, but it's unproven in solid tumors. Certainly in leukemia, this will become increasingly standard of care and be taken into upfront therapies over the next 10 to 15 years, I suspect. So what is the pathway to actually taking a concept like this and trying to make a clinical trial out of it, or to create a successful trial? And this is a long and tortuous process, which I'm going to try and share with you, or at least it's been a long and tortuous process for us. So the initial stage, back in 2004, was to start writing grant applications to do this in neuroblastoma targeting GD2 in the form of project grants to at least start getting laboratory data. Then it's a 2 to 3 year project to actually do all the stuff in the lab to show proof of principle it can work. Then applications for clinical trial grants comes after that, which can be a long process, of course. And then once getting funding, we now then have to take our clinical protocol and actually scale that up and get all the manufacturing aspects of it right. We then have to actually generate a clinical product, something which can be used in an actual patient, which involves a process called GMP, or good manufacturing practice, which has a whole level of regulation associated with it, which is actually extremely expensive to do. And there's not a lot of academic expertise, especially in the UK, for taking things in an academic setting to GMP level and actually putting them into patients. Then there's formal safety testing and scale-up procedures, which have to be done before an application for a regulatory approval of a clinical trial can be put in. So the whole thing can take, as you can imagine, quite a long time. I'll just sort of run through the time scale of how this has been done. So with my co-investigator, Martin Poulet, we wrote a grant application in 2005 to the Neuroblastoma Society, and were awarded a 3-year project to develop the receptor which he had used in that initial study in Baylor, and to try and improve it. And spent 3 years essentially humanizing it and optimizing it and changing it to make it more effective, using a postdoctoral scientist who was employed on that grant. So in 2009, we were in a position we had the data with which we could write a grant for substantial funding actually to develop and run a clinical trial. We approached the Medical Research Council and Cancer Research UK, and were successful to get CR UK support, but it actually took a year in order to go through the processes of applications and approvals and reiterations of the application. And CR UK agreed, using a milestone approach, to fund all our preclinical evaluations and to fund the generation of a clinical grade product, and then to fund the actual clinical trial and oversee the clinical trial if it was successful. So actually for a CR UK, it's an expenditure of about £2 million to do that. We had to generate a trial team, so 3 full-time scientists who've essentially worked on this for the last 3 years in terms of the laboratory aspects of it. There are 4 people within CR UK who form our management team and work on it half-time. And then we've needed to obtain the money for additional strengthening of our hospital research team in order to look after the patients when the trial finally opens. And then myself and my co-investigators, Martin Poulet, Karen Stratoff, have worked on this as one of our main projects for the last 5 years. So the clinical team we have to train to do GMP. So this is how you do GMP. We're fortunate at Great Ormond Street to have our own GMP labs, which were developed for the initial gene therapy trials for immunodeficiency over the last 10 years. But there is a whole range of sterility and regulatory aspects to actually how you do these things. And as you can see, you have to work within isolators using these spacesuits. Then the same people, we've been doing our preclinical. As you can see, we have, in academic university, we are short of cash. We can't afford extra stools. And then there's all these optimization steps required, which we've gone through. What type of blood product shall we use? What is the method for activating the cells? What's the best method for freezing them? Should we do this in plates or in bags? What type of media? What type of serum? Et cetera, et cetera, et cetera. So we've gone through all these things over the last couple of years, and this is the effect of it. So complete exhaustion. Fortunately, our T cells aren't exhausted, but only our members of staff. So this is the only bit of data I'm going to show because it's kind of would be like 50 slides, but this is our final 3. SU stands for scale-up. These are our 3 clinical grade scale-ups showing the expansion of the cells, and this population here is what we're aiming for. Around about 60% of our products are very brightly expressing the chimeric antigen receptor. So that's where we're up to. We've completed our scale-ups. And this is the protocol. This is how the trial will work. We've decided we're going to use fresh apheresis product. So this is not GCSF mobilized. This is just standard apheresis, which many will be familiar with. It's similar to the procedure for stem cells, except we use a vascular rather than a line in the arm, as shown here. And these get the clinical grade virus added to them in a bag, go through this procedure of getting a pure product, and then the final product gets frozen down, goes through the safety testing, which only takes a couple of days, and then the patient undergoes chemotherapy conditioning prior to the injection of the genetically engineered CAR-positive T cells. Okay. I'll come on to how much chemotherapy, because it's a little bit a part of a trial design. So one of the key factors in CAR adoptive immunotherapy is whether you need to give chemotherapy prior to injecting the T cells. Almost certainly you do, I think. And so we will have cohorts of patients. The first group of patients will receive no chemotherapy and just get their own T cells back. The second group of 3 patients will receive cyclophosphamide, and the third group will get cyclophosphamide and fludarabine. The dose of those drugs is moderate, so they will cause a drop in blood count for about 2 weeks. So it's a little bit stronger than a standard dose of chemotherapy you might use, say, in Kojak induction, but it's a one-off, and it's much less than busulfan melphalan. In the first group of patients, if they don't get a response or they don't get expansion of their T cells, they will be able to have a second dose within the second cohort, so that there's no disadvantage to being in the first cohort. Now, initially, it'll be just at our own hospital, Great Ormond Street, because of some of the safety concerns required. So within the development of this area, there's a number of international developments. There are different flavors of these CARs, as I showed at the beginning. We're using one which we generate ourselves, which is unique. It's different to one which was used at Baylor and different to ones which are currently in trials in the States. There's a trial just opened in Seattle with Julie Park, Mike Jensen as investigators, using a different antigen called L1-CAM. And there's 2 trials, one at Baylor, Texas Children's Hospital for neuroblastoma, using a GD2 CAR, but a different type. It's a different generation, and it's got a different antibody part. And exactly the same CAR is also being used in the NCI in Bethesda with Crystal Mackle, but for non-neuroblastoma GD2 positive children's cancers, such as sarcomas. And those trials are open recently. Now, just for 2 more slides, I think, I'm going to talk about what other approaches there are to immunotherapy using this sort of technology. Now, I've shown in here at the top, we've got these red stars. These are antigens, or my depiction of an antigen. So in this slide, the red star is GD2. And I've shown the CAR approach, that the CAR binds GD2 and signals directly to the T cell, or the killer cell, whatever cell you choose to engineer the CAR in. And on the left, I've shown an antibody, like chimeric 1418, binding GD2, and then being recognized by something called a receptor on the surface of the killer cell. And we think that for chimeric 1418 immunotherapy, that killer cell is usually something called a natural killer cell, but other types of T cell can also do it, such as gamma delta cells, as Daniel mentioned earlier. So what variations of this are possible in the future? Well, one is, as I've shown there, we can use different flavors of antigen, as shown by the different colored stars. And these are some of the things, possible future antigens, which are in development in labs around the world, including our own lab. And we're particularly interested in ALK, O-acetyl GD2, something called B7H3, which is the target of 8H9, the antibody Shaquille talked about yesterday, used for the CNS relapse of neuroblastoma. PDL1, which a number of labs are interested in, NCAM and L1CAM, but the list could be much longer. And Holger, in fact, put up a slide, John Maris put up a slide yesterday showing 15 possible neuroblastoma antigens. So this is an area of investigation, and some of these may be better than GD2, in fact, may be associated with less toxicity. So how are we going to fund the future of immunotherapy? So this is a UK perspective on how UK academics go about getting funding for research and the sort of timescales in order to take things from a basic idea to the clinic. And I thought I'd put this slide on because we kind of touched on this yesterday a little bit, especially when Shaquille talked about philanthropy and the involvement of charities in raising money for research. So the most common way of generating funds within the UK is through charities and research councils and grant applications. And that process normally involves about 3 months of writing a grant application. And the current success rate for charities and research councils in the UK for grants is about 20%. So 80% of the grants I write will be rejected, which you could say is meaning each year I waste, if I write 10 grants and it takes 3 months, I waste about 30 months of time a year in writing grants which never... Now, that's not true, because actually the process of writing a grant is a very iterative thing. It forces you to think of the key questions, justify it, and it really puts a very high level of scrutiny on your idea before you ever get any money to do anything. Now, 10 years ago, if you'd asked me, how should you raise money, I'd say, you should just give it to me, because then I wouldn't have to write grants. It's taken me 10 years to get any good at writing grants, and now I can see the value in it. Because when we start a project, we know what we're going to do, and we have the confidence that a lot of other people have looked at it and scrutinized it. Now, if you get industry-funded research, there's often a partnership, and there's often an equally high level of scrutiny involved. The soft money or individual donors philanthropy definitely has a place in pushing the agenda forward, but we must be aware that with philanthropy there's a danger, not a danger, it's sort of a risk or a weakness, that it lacks a level of scrutiny which actually increases standards overall. There can be long delays to taking things forward because we have to recruit people, which takes a long time. We need to set up project management. And then, once we've got the preclinical data, we have to write trial applications. So without doubt, the whole thing can be sped up, but there's a lot to be said also for the grant process and the scrutiny process in terms of ensuring the quality. So I thought I'd throw this out as just a sort of discussion point or an idea of how we take this sort of research forward with the engagement of parents groups.